So today we're going to jump headlong into the first part of our cell cycle. Now when we talk about cell cycle, we're really talking about cell division. And you can see this graphic on the right. We break up a cell's life cycle into essentially 4 phases. We have our first phase we're going to start with here, G1, which means growth phase 1. The cell's just growing, doing its thing. But then we get to the middle phase, which is S, which stands for synthesis. DNA synthesis, then we go through a growth 2, and the M phase, that is mitosis. And mitosis literally means division of the chromosomes. And so we're going to divide the nucleus and the chromosomes, and we're going to divide the cell. So we have karyokinesis, which is division of the nucleus, and cytokinesis, which is division of the cell. That's all going to be happening in mitosis.
What did you say G2 was?
It's just the second growth phase.
Oh, okay.
Yeah, we're going to get in a lot more detail. We're just starting out with some of the basics. But what we're going to do for this first part, 19.1, is we're going to spend our time in the DNA synthesis phase. We're going to see how do we replicate our DNA strands so that each daughter cell receives one original strand from the parent and a newly created strand. And so that's going to be what we're going to do in this first part, 19.1. So here we have, other than mitosis, the remainder of our cycle, G1, S, and G2, collectively together we call interphase. It's between mitotic events. And then here, the M phase, mitosis, which literally karyokinesis, division of the nucleus, and cytokinesis, where we're going to actually divide the cells in half. And we'll end up with mitosis. We'll have 2 daughter cells from one parent cell. And compared to the parent, how are those daughter cells going to look? Identical. So mitosis, in a sense, is cloning. The cell clones itself. You're going to start out with human cell. We're going to start out with 46 chromosomes. That's our 23 pair. And our daughter cells are going to end up with exactly the same 46 chromosomes, 23 pairs. And for all intents and purposes, those chromosomes are going to be identical to the parent, unless we have a mutation, which is fairly rare when we go through cell division. Now again, in our graphic, we're going to have a number of stages of mitosis. The last stage actually overlaps with our cytokinesis. And as we get into 19.2, that second half, we'll go through the characteristics of the individual stages of mitosis. But first, DNA replication. That's what today is all about. Our DNA is double-stranded, right? Watson, Crick, Franklin, they're the ones that deduced that double helical structure. We know that our strands are held together by nucleotide pairs. And what bonds hold them together? Hydrogen bonds, the easiest bonds to break. So as we begin our DNA replication, the first thing we have to do is we have to open up our double helix so that the enzymes that are going to be responsible for replicating a new strand can gain access to the original strand, which is going to be the code from which those enzymes are going to read the sequences and build the new strand. Do you see why it's important that each cell gets a parent strand? Since that cell lived long enough to divide, those strands must be accurate. They must be good. So you want to build the new strands on that good code. Now first, like we've done many times, start simple and get more complex. First, we're going to look at our circular DNA. Now, which cells have circular chromosomes? Bacteria, so our prokaryotic cells. So when we look at our bacterial cell, we have our original template. And what's going to happen is we're going to separate those strands. And you can see we have these strands, and at each end of this separation, we're calling these a replication fork. Because if you take your double helical strand, like I have my fingers together, when you separate them out, it's almost like you have these two prongs. That's where they're getting this idea of a fork. And so you're going to have one on the left and one on the right. And so our replication of our new strand is going to be going in two opposite directions depending on the strand. Because when we talk about directionality of our DNA strands, one strand we said was parallel, 5' to 3', but what was the other strand? 3' to 5'. We call that anti-parallel because it's opposite direction. So we're going to have one strand being synthesized to the right and one strand being synthesized to the left. And in that way, we will end up at the end of the process two circular double helical DNA chromosomes. And so that's how our prokaryotic cells do that. So now we're going to sort of stop motion animation as we go through a movie. Imagine these next few slides being just frames of our video. Here we have our two-stranded circular chromosome. Where we separate the two strands, we're going to call this the origin of replication, or the ORI. ORI is usually how that's abbreviated. Now, would you predict that the origin of replication, is that going to be rich in AT pairs or rich in GC pairs? Why? Why AT? Easier to break. We have 2 hydrogen bonds per pair rather than 3. So that makes sense. So you see, even we have to use our information and knowledge about our purines and pyrimidines to help us understand replication begins. So at our AT-rich ORI, we're going to form a bubble. And again, this is facilitated by enzymes, a collection of them. And we're going to start naming them as we get into our eukaryotic replication. But we have enzymes that unzip the DNA. There you can see our right and left replication forks. We begin to create brand new DNA, which is encoded in yellow. And as the enzyme continues to unzip and we make new strands, more and more, we finally get to the point that we are going to have, and even though there are different shapes here, we have two exact copies ended up with our two brand new circular chromosomes. But notice they all have one original strand and one brand new synthesized strand. That's called semi-conservative replication. And it's the safest way to prevent mistakes and errors as you're making a copy. The biggest part of this is understanding that you're synthesizing the strands in opposite directions, because what is our reading frame of DNA always? 5' to 3'. And when you have one template going in one direction and one in the other, you've got to synthesize the two strands in opposite directions. So it's a bidirectional kind of replication. So when we go to our eukaryotics and we look at our linear DNA, we're going to have a number of places where we have our origins of replication. Again, these are going to be AT-rich, just like we saw with our bacterial DNA. And we're going to be using enzymes that are going to help form these replication bubbles. And notice it's not just one per chromosome. It doesn't just start at one end and go to the next. There's going to be several. And we're going to end up with several pieces of newly synthesized DNA along the length of the chromosome that have to be glued together. And so the enzyme that's largely responsible for unzipping our DNA is DNA helicase. That's going to be one of the many players that are critical in producing our new strands of DNA. So we know we need helicase to unzip, but the other partners we're going to need a couple of DNA polymerases. Now we know that an enzyme usually ends in what 3 letters? ASE. So if you see DNA polymerase, what can you predict that enzyme is going to do? take nucleotides, our monomers, polymerize them into DNA. It's going to assemble a new strand. So we have the enzyme that's going to take our nucleotides. These are our nucleotide triphosphates. And they're going to assemble them together. And they're going to assemble them in the correct sequence, reading the template strand. So I'm DNA polymerase. I'm reading the template. If I am on a C, what nucleotide am I going to pull out of my pouch and assemble into the new strand? A G. Here's a T. Here's a T. A. Here's a C. You see? That's all it is. And if you've ever copied, what?
You said, do you see? And I said, G.
Okay. Oh, C and G. I see what you did there. It's like copying a friend's notes. If you miss class and you have to copy the notes, you know it's not a lot of brain power that goes into that. You're just simply looking at what's there and making a copy. That's what polymerase is doing. It does it fairly quickly, and it takes a lot of nucleotides to assemble into our chromosomes. So again, as we're reading, we are reading that 5' to 3'. And so that's how we're going to assemble our DNA molecule. That's why the two strands, the two templates in two different directions, the synthesis of our new strands are going to be in two different directions. But we still have that number 5' to 3'. And remember, that's going to be where we see the phosphate groups. So we're always going to start with that 5' phosphate, and we're going to have an open hydroxyl group at the 3' end. That's going to help us assemble the nucleotides in the right orientation, in the right sequence according to the template. So far, not a lot of brand new information. So far, just going right along with what we know about with DNA. And when we look at our linear chromosome, it's not going to be a lot different than we see with the bacterial circular one, especially when we look at one replication fork, but we're going to start adding the players and the enzymes and the steps that are involved in forming our new strand. So again, replication origins, AT-rich regions, several of them. We begin making our new strands of DNA in two directions, depending on the direction of the template. And when you put it together, this is what it looks like. Notice we have one strand at the top that is forming just in one continuous long red line. Do you see that? Because if you notice, we're synthesizing the new strand in the direction in which we're unzipping the DNA. So it's just moving along following helicase, helicase being the enzyme that unzips. This is like following an ambulance, right? Are you ever going to have to stop if you're following an ambulance? No. If you ever have a loved one in the ambulance, you know you're just right on their bumper, you're taking them on until a cop tries to figure out what you're doing, right? But notice the bottom strand. It's being synthesized in the opposite direction. So if you notice, we make a strand, then we make another strand, and this is called the lagging strand or the discontinuous strand. So you see, we're having to create the DNA in sort of two different steps and two different mechanisms, even though when we finish, we're still going to have new DNA strands that look the same, that are encoded for by our blue templated strands. Now, two pieces of information we're going to introduce here that we'll expand on in later slides. Notice the little green segments that you see. Do you see the green? The green are primers. DNA polymerase cannot synthesize a brand new strand and connect nucleotides to thin air. So DNA can't read that first nucleotide and say, okay, here's an A, I'm going to put a T down. This is the very first one in my new strand. DNA polymerase can't do that. DNA polymerase, however, can attach nucleotides to an existing strand. But oddly enough, the primer, the initial beginning of the new strand, is laid down by another enzyme we're going to introduce in a little bit But the primer is RNA. It's a little short piece of RNA. And it's from that RNA that DNA polymerase then extends the new strand. And so at our leading strand, that's not a big problem. You start it with a little piece of RNA and you can just keep going until you run into the next strand that was produced at that next origin of replication. But in our lagging strand, notice we make all of these little small fragments. See, there's a fragment, there's a fragment, there's... Those fragments are called Okazaki fragments. And notice what happens to our little green, what happens to our green RNA? It's going away. So even though for both strands we use RNA primer to start the extension, the RNA has to be removed, and we're going to have an enzyme that does that. We're going to have DNA to fill in the space where the RNA primer was. We've got an enzyme that does that. And then we need an enzyme that's going to be able to glue all of these fragments together. And we have an enzyme that does that. So that's an overview of what we're doing here with our DNA replication. And I think now we get to have a little bit of amoeba sister fun.
So here we go with our key players. First, helicase. This is the unzipping enzyme. If you recall that DNA has two strands, you can think of helicase as unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, poor DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer that it makes? It's actually a piece of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that a little later. Now don't feel overwhelmed. We'll go over this sequence in order. Please keep in mind that like all of our videos, we tend to give the big picture, but there is always more detail to every biological process we cover. That's really true for DNA replication. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. There can be multiple origins within the DNA. At the origin, helicase, the unzipping enzyme, comes in and unwinds, unzips, the DNA. SSB proteins, that stands for single-stranded binding proteins, bind to the DNA strands and they keep them separated. Primase comes in and makes RNA primers on both strands. Now remember, that's really important because otherwise when DNA polymerase comes in, it wouldn't know where to start. Now DNA polymerase can get to work. Remember, it's the important enzyme that adds DNA bases. Now, you have two strands, right? They're not identical. Remember, they complement each other. They also are anti-parallel, so that means they don't really go in the same direction. With DNA, we don't say a strand goes north or south. The directions for the DNA strands are a little different. We say that DNA either goes 5' to 3' or 3' to 5'. This can seem very confusing. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of the ring structure. Now you do the same thing for the other side, but keep in mind this strand is flipped, just because DNA strands are anti-parallel to each other. So let's count these. Again, clockwise from the oxygen, 1', 2', 3', 4', 5', and the 5' is out of the ring. This strand on the left runs 5' to 3', and the strand on the right here runs 3' to 5'. Well, it turns out that DNA polymerase can only work in the 5' to 3' direction. So the strand that runs 5' to 3' is fine. It's called the leading strand. But the other strand will make it a little tricky. DNA polymerase can only go in the 5' to 3' direction. So RNA primase, it has to set a lot of extra primers down to do that, as shown here. It takes longer too. This strand is called the lagging strand, which is pretty fitting. On the lagging strand, you tend to get these little fragments of synthesized DNA. They're called Okazaki fragments. Amazing name. The primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, remember that's the gluing enzyme as I like to nickname it, has to take care of the gaps in the Okazaki fragments. Now at the end, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call this semi-conservative replication because the two copies each contain one old original strand and one newly made one. Oh, one last thing. Surely you have had to proofread your work before to catch errors. Well, you definitely don't want DNA polymerase to make an error. If it makes the wrong DNA base, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein, or no protein. DNA polymerase is just awesome. It has proofreading ability, which means it rarely makes mistakes. And that's a good thing. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, now let's just look at it in our detail. And we can go through these fairly quickly now simply because we saw what was going on in the little cartoon. So DNA helicase, unzipase, unwinds the DNA. Now, did you notice that the Amoeba Sister talked about single-stranded binding proteins? So as helicase unzips these proteins, SSB, single-stranded DNA binding proteins, they bind so the DNA can't get back together. Otherwise, as helicase moved off, the DNA would just re-anneal together. So the single-stranded binding proteins keep them as single-stranded long enough to begin the replication process. Now, as helicase is unzipping, it creates tension in front of the chromosome, more supercoiling. So what enzyme do we know releases supercoiling? Topoisomerase. We learned about topoisomerase yesterday. Now we have two different kinds of topoisomerases. Do you remember what their names were? Topoisomerase 1 and 2. What's the difference between 1 and 2? Okay. Topo 1 makes a single-stranded cut. Topo 2 cuts both DNA strands to release the tension. All right, that's as difficult as it is. Topo 1 single-stranded cut, Topo 2 cuts both strands. Hence the numbers. So that prepares us for the replication process. Now, DNA polymerase is going to be essential. If we don't have DNA polymerase, we cannot assemble a new strand. Now, again, there are a lot of other players like primase and ligase as well. But DNA polymerase, we're going to mainly focus on DNA polymerase 1 and DNA polymerase 2. They produce the new strand in the 5' to 3' direction, but they actually read backwards. But as they read, they also have exonuclease activity. Exo means from the outside. Endo means within. And so this is their proofreading activity. So as DNA polymerase is going along, on the template, there's an A. What does DNA polymerase put in the new strand? A T. Here's a C. A G. Here's a G. You put a C. Well, DNA polymerase accidentally drops an A here. And DNA polymerase moves on to the next one, but then DNA polymerase is always checking again. Oops, I got that wrong. So DNA polymerase would clip out that last nucleotide it incorrectly put in, back up, and put the correct one in. See, always checking before it goes on. That's the proofreading activity. And it really minimizes the number of errors that could ever happen. Now here at the top, when we look at our DNA polymerases, they both read 3' to 5'. They both synthesize 5' to 3'. But there's one very big difference between DNA polymerase 1 and DNA polymerase 3. DNA polymerase 1 has the ability to remove and replace RNA primers. So when we first begin the process and primase comes in, produces our short RNA primer, DNA polymerase 3 is going to come in to make our strand, whether it's the leading strand or the Eukaryotic strands. But then DNA polymerase 1 is going to come in, remove the RNA primer, and fill that space with DNA. That's what the difference is going to be between DNA 3 and DNA 1. And that's why we put this here on this chart. There's a lot of other enzymes that you can read here, but on this particular chart, the two in red, those are the things I want you to really pay attention to and what makes them different from one another. Now, there's so many enzymes, the topoisomerases, the helicases, the primases, the two different DNA polymerases, single-stranded. So they call this the replisome. It's like scientists are always looking for cute names to apply to different events and different organizations. So the replisome involves all the proteins and enzymes that are used at the replication fork to carry out the process of producing our two new strands from our two templates in a semi-conservative fashion. We keep saying that semi-conservative because that is a critical aspect of DNA replication. Now, I don't really have a good answer for this, but why do we use RNA as a primer? Why don't we just use DNA as a primer? Well, you've got the two strands together with a primer, so it's not going to re-anneal. It may be that the enzymes that might add DNA in that place don't exist, and we had this primase that can make RNA, and it just does it. It may be that that enzyme is more critical of the nucleotides it assembles, so it's going to be even fewer errors than another enzyme. Regardless of the actual why, we know that that's how it happens. And primase, when it binds with the DNA and other proteins that are involved, that's going to be a primosome. And so that primosome is going to sit down, it is going to form our RNA piece, and then from that RNA, now our DNA polymerase 3 is going to be able to extend the new DNA strand in a polymerization event. But then after we synthesize the strand, notice here's a new strand coming. Now this could be either leading or lagging strand. If it's lagging strand, these are two Okazaki fragments, 1,000 to 2,000 bases. But if it's our leading strand, it's going to build a strand until it reaches the next origin of replication strand, and that still is going to have to be glued together. We still have to remove a piece of RNA, and that's where DNA polymerase 1 comes in, sits, removes the RNA piece, it synthesizes a new piece of DNA, and there's still going to be a gap. We filled it in here, and this illustration doesn't show it, but what fills in the gap is an enzyme called DNA ligase. So like the amoeba sisters showed, primase, polymerase, helicase, and ligase are 4 critical enzymes that are going to be used for DNA replication. Topoisomerase is going to be in there to remove the supercoiling, but those 4 players in our little video, those are the critical players for DNA replication. So let's step through the process. Here are strands of DNA. We have a strand that is called the sense strand, and this contains the sequence that's going to be identical to the sequence of our messenger RNA. This is the actual code we're going to use to build the proteins. Since the opposite strand is complementary and anti-parallel, it's sometimes referred to as the antisense strand. So you see, this is the strand, the antisense strand is the one that we're going to read from to make our messenger RNA that's going to be identical to the sense strand. The sense strand is going to be a template to make a new antisense strand in DNA replication. All right? And notice the direction, 5', 3', and 5' to 3'. So first thing, helicase comes in, unzips. Again, we're at the origin of replication. We're in the replisome. And as we're talking about leading strand synthesis, we're going to assume we've already got a primase and an RNA primer somewhere off the screen, because here comes DNA polymerase. What number would you put with this DNA polymerase that's extending the strand? DNA polymerase which? 3. Now, you're going to be inclined to say 1 because it's the first one that's doing the first thing, but actually 3 is the one that's going to extend the strand. 1 is going to be the one that removes the RNA and adds in those little pieces. So DNA polymerase 3 comes in, building our new 5' to 3' strand, and it's following right along with DNA helicase as it's moving to the right. That's our leading strand. But now we're going to focus on the lagging strand. The lagging strand is going to be different because its polymerase is synthesizing in the opposite direction with which helicase is unzipping. So we call this discontinuous replication. And we're going to have a number of priming events with RNA primer. We're going to have a number of shorter fragments called Okazaki fragments. And again, for eukaryotes, we're talking about as short as 100 bases. For our bacteria, maybe 10 times, 20 times longer. So here we've already made our leading strand. Now we're going to do our lagging strand off the top. And what's the first enzyme? We've got our single-stranded binding proteins. I haven't put these in here because that would just muddy up the picture. But what is the first enzyme that has to come in before we can start making our new strand? The RNA primer, so that's primase. So the primase enzyme comes in as the primazone with all of its other partners. It attaches. It makes its RNA primer, which in this case we've illustrated in red. Then primase pops off, and here comes DNA polymerase 3 that's going to extend the strand. Yellow is our new strand of DNA, our Okazaki fragment that's only associated with the lagging strand. So we've made our Okazaki fragment. Now helicase is continuing to move away to the right, which opens up more single-stranded DNA at this point. So what do we do now? Well, we get another primer coming in, lay down our RNA primer, and DNA polymerase 3 makes our new Okazaki fragment. But do you see now we've got RNA interrupting our DNA strand. And we've got a break between these two Okazaki fragments. So how do we remove and replace the RNA? DNA polymerase 1. It comes in, attaches, removes the RNA, lays down new DNA in its place. But what do we still have? We still have a gap. We still have two separate Okazaki strands, and we have to glue them together. What enzyme is the gluing enzyme? DNA ligase now comes in, attaches, and glues the strands together. This is happening all along the length. So basically, one strand's just chugging along, the other strand is discontinuous, being interrupted, but at the end of the process, you're going to have two brand new complete strands that were assembled in different directions yet are complete and accurate. because of the proofreading capability of DNA polymerase. So here we go, just stepping through it. And these are happening at the same time, by the way. So your leading strand, the lagging strand with our 2 Okazakis, replacing our RNA, ligating it together. And again, we're going to have another event removing the second RNA molecule by polymerase 1, filling it in, ligase, and doing that again and again for the whole length of the strand.
Is chromosome only associated with the lagging strand?
No, we had that kind of off the screen here. No, the primosome and primase is going to always be used for the initiation of a new strand in leading and lagging.
Those 2 words interchangeable.
Primosome is the whole thing, the whole complex of a bunch of different enzymes. Primase is the one that actually lays down the RNA primer.
Okay.
Right. Now you're going to have many, many more RNA primers in the lagging strand and many fewer in the leading strand. Because if the Okazakis are 100 bases long, the leading strand is going to be thousands of bases long. That'll be the only difference. But it's a good question because it's not in this little cartoon, not in this little illustration. So again, we talked about the proofreading ability. DNA polymerase 1, DNA polymerase 3, they can correct errors that they just made in the assembly of the chain. And when you look at the mistake rate of our DNA polymerases, they're quite low. Let's see, I wanted to get this slide. So we typically are going to produce an error every 1 million bases. Now that's pretty good. If you do something 1 million times in a row and you only have one mistake, that's a pretty good day, right? But when you look at the proofreading, that improves the mistake from 1 million to 1 billion. One mistake in 1 billion bases. So essentially, you're not going to make a mistake. Because if you make a mistake, what do you end up with? A mutation. Mutations can lead to what horrific disease? Cancer. Now, if you have that in your body cells, yeah, that's going to be cancer. If you make a mistake when you're making your copies of DNA and that mistake ends up in your sperm or your egg, that's going to get passed on to every cell in the new individual, in your child. So those are often very rare. Mistakes that happen in the chromosomes usually lead to spontaneous abortions so that the mother never even knew she was pregnant. But you want to protect your germ cells at all costs. One of the things that you really have to think about as far as protecting yourself and your germ cells is when you're getting x-rayed. At the dentist, if you're going to the doctor, make sure that they're covering your pelvis with a lead apron if you're having an x-ray for anything whatsoever. Because those x-rays are penetrating through your body and they can cause DNA damage that could be passed on. You don't want that to happen. So this is just a static summary slide showing our helicase. They call this gyrase. That's another name for topoisomerase. Our little cheese wedge, that's our helicase. Single-stranded binding proteins, leading strand along the bottom with DNA polymerase 3, lagging strand along the top. You can see our red RNA primers, DNA polymerase 1 coming in, removing and filling in, and then DNA ligase there at the bottom, the little purple hourglass, sealing them off. Now, DNA ligase is also going to work on our leading strand. It's going to join one leading strand with the next leading strand at the next origin of replication. So, but just like the primers, there are many fewer ligation events in the leading strand for an entire chromosome. So we talked about X-rays. Well, that's a form of radiation. And sometimes you can form thymine dimers. Now, these thymine dimers meaning you're going to join two thymines together and you can't get them apart. If you can't unzip your DNA, you'll never replicate the DNA. We can also have chemicals that get in between our nucleotides and insert it, and that can prevent the unzipping of the DNA. It prevents the replication of the DNA, which is going to lead to mutations. But we can have some that just happen, just with age. You can lose a purine, which is a loss of an A or a G, or deamination that changes a cytosine to a uracil. Now, where are you ever going to find uracil in DNA? You shouldn't. Where's the uracil? It's in RNA. Now, this deamination is going to be important when you get to physiology, because that spontaneous event happens in your genes for your antibodies. And it causes your antibodies basically to produce random sequences so that your body can produce antibodies that practically would recognize any possible antigen you may encounter. But it happens just random happenstance because of the deamination of some of the cytosines to uracil and then how it's repaired. In your immune system cells, you have a polymerase that I call the stupid polymerase, or the hourly worker polymerase. Because when it sees that uracil that happens spontaneously, it comes in with its proofreading capability and says, wait, there's a uracil. I don't know what to do with that. I could call the supervisor, but that's going to take 10 minutes, and I go on break in 5. So it just reaches and gets a nucleotide and just drops it down and keeps going. So that spontaneous deamination leads to a random placement of one of your 4 nucleotides. And it's that shuffling that provides you with an infinite variety of antibodies, but using only a few genes in your genome. Now, that's a whole section of immunology that we'll talk about when we get to that point. So with our DNA repair, certainly we talked about the exonuclease activity of our polymerases. Well, we have certain repair enzymes that can come in. They detect that damage because you have a mismatch. You know, if you're trying to put a cytosine with a thymine, it's not going to work. You might have the space too far apart or not close enough together. And so our endonucleases come in and they remove and cut out that piece. Well, once we've got that piece cut out, we've got some DNA polymerases come in, fill in that gap, DNA polymerase 3, because it can extend from DNA. It can also extend from RNA. And once DNA pol 3 comes in, makes the new strand, DNA ligase comes in, boom, fixed. Now, in many instances, we call these sort of endonuclease activities, we call these tumor suppressor genes. Have you ever heard of BRCA1 and BRCA2? BRCA1, BRCA2, breast cancer early onset gene 1 and gene 2. This is the gene that many individuals get tested for, and if you have a mutation in BRCA1 and or BRCA2, you could be at 75% greater risk of developing breast cancer. This is the gene that was mutated in Angelina Jolie, and she just decided to have radical bilateral mastectomy rather than run the risk of it. So that's what a normal BRCA1 or BRCA2 would do, would find the mismatch, repairs, and fix it. Do you see how if you lose the ability to repair your DNA, there's a greater risk of a mutation being sustained and that mutation being in the wrong place to lead to cancer? So there are a number of these proteins, these enzymes that are endonucleases, BRCA1, BRCA2, only being 2 of them. But it's the one that I thought you may have heard about, you know, in the news and in the media.